Hi guys, welcome to Lab 2.1, Cell Organelles. Today we're going to be using a compound microscope to look at different examples of cells. The first thing that we're going to do is get our microscope out and make sure that our stage is lowered all the way down, and we are using the scanning lens, which is the red lens. So today we are going to be looking at a slide of a living model, which is going to be our cheek cells from my mouth. And then we are going to identify parts of a cell and then compare that to an animal and a plant cell. Okay, we're also going to learn about the functions of each of those cells. So number 1 says take a clean toothpick and gently scrape the inside of your mouth. So what I'm going to do is just take a toothpick and gently swab the inside of my mouth with that. Next, I'm going to take a slide and just place it on my table right here. And I'm going to need a slide cover, so I'm going to get that ready. Now I have thoroughly swabbed the inside of my cheek with this toothpick. And I'm just going to smear it right in the middle. It's kind of hard to see, but there are going to be cells that have come off of my mouth and the toothpick, so I'm going to be able to see both. The next thing I'm going to do is take methylene blue. Methylene blue is a dye that is really electropositive. So what it's going to do is grab onto anything that is electronegative. And the methylene blue is a very, very strong dye, so it can get on your hands and clothes, so it's best to be very gentle with it. And I only need a very, very, very small drop. I don't need a big drop. So now I'm going to put that to the side. The last thing that I'm going to do is, using my slanting technique, I'm going to gently put a slide cover over the top. And you see how dark blue that is? I don't want it to be that dark, so I do need to get a paper towel and just gently place the paper towel folded over the top and gently blot. I don't need to press down. I'm not putting any force on it. I'm just blotting it, and you see how all that excess methylene blue came off. And now I have a prepared slide. Okay, so I will place this underneath the slide clip. And now I'm going to use my X and Y axis to be able to move the slide right over the center of the light. Now, I'm going to look through the microscope and move this large knob, the coarse focus, forward until the specimen comes into view. I'm only using the large knob and the scanning lens at this moment. Okay, give me a minute. I'm working on the view here. Okay, now that I can see my sample, all those blue little flakes and specks, that is all epithelial cells or cheek cells. Okay, so once I have them in focus, I'm going to turn to the low-powered lens. I didn't change anything. I didn't refocus. I didn't turn any knobs. I just changed from one lens to another. Okay, I'm moving my X and Y axis to get a better view of a clump of cells. And now I'm only going to use my fine focus. I'm not going to use the coarse focus anymore. I can kind of move it out and in to get a really sharp image. Okay, and now I'm going to switch to high power. All right, here we are in high power, and I can see each individual cell. They're very overlapped, okay? Epithelial cells do overlap. So what I'm gonna do is I'm gonna try to find one kind of by itself that might be a little bit easier to see, like this one, this little guy right here. So what I'm looking at is going to be the cell. Inside the cell is the cytoplasm that's light blue. The border of the cell is this plasma membrane made up of phospholipids. And in the center of the cell is the nucleus, and it looks dark blue. All those little dots and specks are going to be clumps of bacteria. Now, this specimen appears blue because it's accepted the dye, and the dye kind of sticks inside of the cytoplasm because of that plasma membrane, because of that phospholipid bilayer. Okay? The nucleus appears very dark because the nucleus is very electronegative. DNA is very electronegative. And so the DNA appears dark blue because it accepts most of the dye. So at the bottom of our page, we have these questions. It says list all the organelles you can identify. Well, the main one we can identify is the nucleus. All these little dark specks are going to be bacteria, so they're not actually part of the cell, they're just next to it. The nucleus is the only organelle that I see other than the plasma membrane. So that's the answer to number 1. And we can see the nucleus and not the other cell parts because the other cell parts are continuous with the cytoplasm. They're indistinguishable from the cytoplasm. They're all kind of made of the same stuff, have about the same electronegativity, and so it looks empty even though there are a lot of things packed into this cell. The inside of the cell is blue because it's accepted that methylene blue dye. Let's move to the next page. The next page is asking me to view an animal cell and a plant cell under the microscope. Here I have a prepared slide of an animal cell and a plant cell. So let's do the animal cell first. All I'm going to do is take this sample and put it under the microscope where my sample is, okay? I'm going to follow focusing techniques to be able to view this cell under the microscope. And remember, we always start with the scanning lens to be able to get in focus. So we always start with the scanning lens. Okay, so now we have the cell in focus, but you see that circle, that is not a cell by itself. That is a blastocyst. And so it is made up of a bunch of different cells. So now what I'm going to do is switch to the low-powered lens, and I'm going to refocus until I can really see those nuclei. And now I'm going to switch to the high-powered lens, and now I can see the cells individually. And the cool thing about this is that I can actually see some cells that are undergoing mitosis. Like this one right here is undergoing mitosis. You see how it has a straight line as opposed to chromatin, which would be all jumbled together. And I apologize, the camera has a really hard time focusing down the microscope tube. I had better luck on this side. Let me see if I can do it on this side. The autofocus seems to catch the inside of the lens instead of the image. Well, I could see them. There we go. All right. So now we can see the autofocus is frustrating me. I apologize. There we go. Okay, so we can see the individual cells and their nuclei. Now what I want to do is switch to a plant cell. Okay, these are my plant cells, and I am going to switch to the higher power lens and refocus. If my autofocus would catch, there we go. And I can go even further. And now I can see the individual stroma, which are part of the underside of a leaf. I have a different kind of plant cell to view though. It's called an onion root tip. Okay, and the onion root tip, you can see the nucleus of the plant cell. So let's look back at our graph. So here it wants you to draw an animal cell. When we looked at the animal cell, we could see it was mostly circular, but they kind of overlapped, and they all had a region, a darker region, which was the nucleus. The plant cell, however, was mostly organized in this square, rectangular pattern, and it had a very large nucleus region. So next we're going to compare and contrast the characteristics that we saw in the animal cell versus the plant cell. For instance, the shape of the cell. In the animal cell, it was circular. In the plant cell, it was rectangular. The cell barrier for an animal cell was the plasma membrane, which is made up of phospholipids. In the plant cell, it was a plasma membrane, but also a cell wall. For cellular energy, animal cells have mitochondria. Whereas plant cells have mitochondria and chloroplast. In animals, we have small vacuoles, whereas in plants, we have one large one. Animal cells are more mobile. They could have a flagellum or cilia, whereas plants are mostly immobile. They do have some moving parts. For instance, the trigger cells in like a Venus flytrap, but it's still not mobile. It's not moving around in a free living environment. Other observations, animal cells are going to have something called a centriole, and they're going to have lysosomes. Whereas plant cells, they're unique in that they have a cell wall, they have chloroplasts, and they have one large vacuole. At the bottom, it's describing cleaning up. So what we're going to do is we're going to take all of our trash that has touched our cells and put it in the biohazard container. So all of this is biohazard waste. And then we're going to make sure to put away our microscope properly. So we always want to pull the stage all the way down. We're going to put our prepared slides back where they belong. So plants with plants, animals with animals. We're going to turn it off, and we will take the cord and put it underneath the stage and then put the dust cover back on top. So we'll do all of those things, and remember, we always carry it by the arm and the base. But right now what I want to go over is the back. If you have your phone, you can scan this QR code to get to a cellular model, and we can use that model to answer these questions. Mitochondria, the purpose or the function of mitochondria is to produce cellular energy in the form of ATP. Ribosomes are to produce proteins. The nucleus is to house the DNA or the genetic material. The endoplasmic reticulum's job is to produce and package the proteins. The Golgi apparatus takes those proteins from the endoplasmic reticulum and further folds and modifies them and sends them outside the cell in a vesicle, a transport vesicle. The protein itself could be an enzyme, it could be a hormone, it could be anything. It could be a cell receptor. The cell membrane is made up of phospholipids and it controls what enters and exits the cell. The lysosome is a special type of vesicle made by the Golgi apparatus that is going to house an enzyme, a digestive enzyme called lysozyme. And it will be it will digest broken parts of the cell or maybe even food vacuoles, and it will break those down into parts that it can then reuse. The last one says nucleolus, but I scratched that out and wrote vesicle because vesicle is more important in this context, in this activity. So vesicle is going to be kind of a little transport bubble. It's made up of phospholipids, and it's usually made of pinched off pieces of the endoplasmic reticulum lumen, the barrier there, or the Golgi apparatus. So here we have a paragraph that's describing Grant City. It says its main export and product is a widget. So according to this, the main export and product is protein. So under the city part for protein, we're going to write widget. Okay. Next, it says everyone in town has something to do with making the widget, and everyone has a job to do. The town hall has the instructions. So if we look down here, the town hall is most similar to the nucleus. Next, it says that the citizens can take the instructions and make their own widgets. Widgets are produced in small shops around the city, but most of these shops are located in the carpenter union. So the small shops here are kind of like ribosomes. And ribosomes can be found in the endoplasmic reticulum, so that's kind of like the carpenter's union. Next, it says after the widget is constructed, they're placed on special carts which deliver the widget. That's kind of like a vesicle. And the widgets are before they can be exported, they're sent to the post office where they're packaged and labeled. That's most like the Golgi apparatus. Next, it says some of them don't really turn out right and they're sent to the scrap yard. They're rejected and sent to the scrap yard to be recycled or broken down for parts. That's kind of like a lysosome. After that, the town has to power the shops, and we need power, and we get the power from a dam. So we're going to write dam next to mitochondria because mitochondria produces ATP. And that dam is going to produce electricity. After we talked about the dam, the entire city is encased by a large fence. Only the postal trucks are allowed to pass. So the cell membrane is most like the fence. Okay, so you can complete this on your own and maybe even think of your own cell analogy. What's another thing that kind of operates like a cell where we have a central town hall and then everybody else has a specific job to do? The next thing that we're going to do is scan this QR code at the back. It is your at-home cell organelle identification. It says use the Cell Alive's website to identify the parts of the cell. Label each part by coloring them on the following coloring page. So what you're going to do is use this as a guide to find out what each of these items are. Once you find out what they are, you're going to write a short definition and then you're going to color it on the next page. So for cell membrane, it says light brown. You're going to come to the next page and you're going to color the cell membrane light brown. At the end, you should have a very colorful diagram that you can use to take notes. And you can even label each of these items by writing at the end of the line. So that's an item that y'all can do at home. Color it with colored pencils, but more importantly, make sure that you write the definitions and you're using this as your guide. Thank you for joining me this week, and I will see you next week.